what is responsible for the dysregulated cell growth? This is another interesting thing. Before we go into the nuclear transfer experiment, mitochondria damage, how's that going to lead to dysregulation? Because the signature phenotype of cancer is cell division out of control. Dysregulated cell growth. How do you get dysregulated cell growth from damage to the mitochondria, right? You have to realize that all of the cells that existed on the planet before oxygen came into the atmosphere were all dysregulated in their growth. There was unbridled proliferation. And they were all using a fermentation mechanism because there was no oxygen yet. So when oxygen came into the atmosphere, it's only then that you began to produce ROS as signaling molecules that allowed metazoans to form. So the differentiated state of our cells is controlled largely by the mitochondria. The mitochondria also control the genes that regulate the cell cycle. So what happens when the mitochondria become no longer operational within the cell, they fall back on their ancient pathway of fermentation, which is linked to dysregulated cell growth. So all this stuff makes real sense if you understand evolutionary biology. What I find in my discussions with students or with physicians, wherever, most people don't even understand evolutionary biology. It's actually a black, it's very hard to convince people of mechanisms when they have no clue about evolution. And believe me, when you see people writing these different kinds of things, you already know they're using teleological explanations, using all this kind of stuff. So once you understand evolutionary biology, you understand the function of these different organelles and how they all came to pass, you can put this whole puzzle together with not too much difficulty. But the nuclear transfer experiments were clearly showing the beauty of these experiments. They were asking, what do gene mutations, how can gene mutations regulate development? But the evidence that they got was that they were actually destroying the somatic mutation theory of cancer by the very observations that they were making in transferring tumor nuclei into normal cytoplasm and normal nuclei into tumor cytoplasm, doing frog work, mouse work, different cancer work, all doing different model systems. And majority of these people coming to a similar conclusion that there's something in the cytoplasm that's playing a bigger role in driving dysregulated cell growth than there was in the nucleus. And when you look at that, I had long discussions with Dr. Kimmel, who did the frog work. He even told me, he showed me pictures of frogs that were developed from the nucleus of a kidney tumor. And he said, frogs can regenerate their tail and limbs and things. And he would cut the tail off this tadpole that was generated from a cancer nucleus. And the tadpole completely regrew the tail exactly as it should have been, despite the fact that this is regulated cell growth from the nucleus of a dysregulated cell. Can you believe this? So clearly it was all in the cytoplasm. Wasn't it? The genes were not controlling the regulation of the growth. So when I told him it's the mitochondria, he was like really excited. He said, yes. He says, yes, you're absolutely correct. It is the mitochondria that doing all this. So that's a hard pill to swallow because you realize most of the stuff in cancer is based on the somatic mutation theory. And if the somatic mutation theory is incorrect, what is the probability of developing new drugs that are going to be really effective if the very foundation upon which you're building these drugs is wrong?